Chapter 2, Neuroscience and Behavior. To begin, I'd like to talk about Albert Einstein's brain. Albert Einstein died of a ruptured abdominal aneurysm in 1955, and pathologist Dr. Thomas Harvey removed the brain upon his death. With the family's consent, Dr. Thomas kept the brain for scientific study. At the time, Dr. Harvey reported that the brain was well within the normal range for size and weight. So, if Einstein's brain was no bigger than the average man's, what did account for his profound intelligence? Well, in 1996, Dr. Sandra Whittleson reported that the inferior parietal lobe was 15% wider than the average person's. Visual spatial recognition, mathematical thought, and imagery of movement are strongly dependent on this region. Einstein's brilliant insights were often the result of visual images that he translated into the language of math so we could all understand it. So, for example, his special theory of relativity was based on his reflections of what it would be like to ride through space on a beam of light. Scientists also reported that the Sylvan fissure, a groove that normally runs through brain tissue, was shorter than average for Einstein. This meant that the cells were packed more closely together, permitting more interconnections and more cross-referencing of information and ideas. However, the most profound difference in Einstein's brain was the significant amount of glial cells in his brain. So, in this chapter, we're going to look at both neurons and glial cells. And remember, a case study is a type of descriptive research that is completed on an exceptional individual with the intent of learning things about the rest of us. The study of Einstein's brain was a great example of a case study that showed us some very important aspects of how we all function. Before we drill into neuroscience, I'd like you to take a moment and watch the brief video that you see on the screen. And this is a video of the brain, and it's narrated by Leonard Nimoy. Now that you've watched that introductory video, let's talk about neurons and synapses. Neurons are also called nerve cells. They are the building blocks of the nervous system. You have three types of neurons: sensory neurons, motor neurons, and interneurons. You also have glial cells, and that's what I was just talking about in Einstein's brain. So in normal people, glial cells actually outnumber neurons by about 10 to 1. Glial is named for the Greek word for glue because it was originally thought that the glial cells were simply the glue that held the more important neurons in place and together in the brain. However, we know that glial cells do much more than simply provide glue. They provide support in nutrition for neurons. You have a special type of glial cell in your brain that is called an astrocyte. And these glial cells are very unusual because it was once thought that only neurons communicate and provide connections to other neurons. However, we now know that glial cells, in some instances, such as in the case of astrocytes, act like neurons. Another type of glial cells supports and forms the myelin sheath, which is a fatty insulating substance wrapped around some neuron axons. So the conclusion that we can draw is that glial cells are much more actively involved in regulating the neuronal communication and activity of the neurons in your brain than previously believed. So now let's look at that first type of neuron, sensory neurons. So, sensory neurons carry messages from the body's tissues and sensory organs inward toward the central nervous system. Motor neurons. Motor neurons lead from the central nervous system to body tissues. Next, you have inner neurons. Inner neurons carry information between other neurons, and the important thing to remember about inner neurons is that they are only found in the brain and the spinal cord. Finally, I'd like to introduce you to another special type of neuron, and this is called a mirror neuron, and these have only recently been discovered by scientists, and they were accidentally discovered when they saw that they were doing research on chimpanzees, and when one chimpanzee was picking up a ball, scientists noticed under a functional MRI that a chimpanzee that was watching the original chimpanzee pick up the ball, his brain lit up the same way as if he was actually picking up a ball. So sometimes scientists concluded from that mirror neurons are one of the building blocks for our ability to feel empathy or to experience what someone else is experiencing without actually experiencing that activity ourselves. So you might experience this, for example, if you're watching a football game and you've played football before and you're watching a football player perhaps throw a great pass and you know what that feels like to throw it. And then you see another player catch it and you know exactly what it feels like to catch that ball. When you know this, your mirror neurons are being activated. I'd like you to pause here and watch this short video on mirror neurons. Now let's look at the parts of a neuron. First of all, let's look at the cell body. The cell body contains the nucleus. It's the round, centrally located structure that contains the DNA. It controls protein manufacturing and directs the cell metabolism. However, it has no direct role in neural signaling, but it does support the entire neuron in the process of neural signaling. Jutting off the cell body, you have dendrites. The dendrites are responsible for receiving messages from other cells, and they conduct that information to the cell body. So the dendrites, in short, are the listeners. We know that generally we are not able to grow new nerve cells in our brains. However, we can create new dendritic connections between nerve cells, and this is the basis for all learning. Hebb's law says that if it fires together, it wires together. In other words, each time you do something new, you are creating a new connection in your brain. And each time you repeat that activity or behavior, you are strengthening that new connection by making even more dendritic connections between neurons. And that explains why the more you do something, the more you practice something, the easier it gets, because you have now more dendritic connections between the neurons that are responsible for that activity. Next, let's look at the axon. The axon is the cell's output structure. You only have one axon per cell, but there are two parts to each axon. So the axon consists of a tube-like structure, but at the very end of the axon there are branches that connect to dendrites of other cells. So the axon extends off of the cell body, The axon passes the message to other neurons, muscles, and glands. The axons are the speakers, and they project several feet through the body. Next, let's look at the myelin sheath. The myelin sheath is the white fatty casing on the axon. It acts as an electrical insulator. But remember, there is not myelin sheath on all axons. When the myelin sheath is present, it increases the speed of the neural signal down the axon. The importance of myelin sheath is evident in the disease multiple sclerosis. MS typically attacks people in early adult life, and it causes muscular weakness, lack of coordination, impairment of vision, and speech. MS attacks the myelin sheath in the brain and the spinal cord, as well as the optical nerve. Magnetic resonant imaging can detect hardening of the myelin sheath on the axons. However, magnetic resonance imaging was only invented in 1973. So prior to that, doctors diagnosed MS by taking advantage of the fact the myelin sheath speeds the axonal conduction. So doctors developed a test using the optic nerve. The doctor stimulated the eye with a checkerboard pattern and then assessed the time it took the impulse to cross the optic nerve by measuring the time it takes for the electrical response to get to the part of the brain that is the target of the optical nerve. The electrical response is measured from the scalp. So people with MS showed significant slowing of the speed of their optic nerve. Now, doctors use the MRI to diagnose MS. But doctors have learned that MS is influenced by both nature and nurture. So the role of the environment is suggested in the cause of MS by studies that show that people who spent their childhood in cooler climates are more likely to develop MS, perhaps as a result of vitamin D deficiency from a lack of exposure to the sun. However, genetic factors are also indicated by the fact that MS is rare in Asians, even when others in their geographical region show a high incidence of MS. Now let's look at how neurons communicate. Neurons communicate by means of an electrical signal that is called the action potential. Action potentials are based on the movement of ions between the outside to the inside of the neuron cell. When an action potential occurs, a molecular message is sent to neighboring neurons. So the action potential is an impulse or an electrical charge that travels inside the axon. And speeds range from 2 miles per hour to 200 plus miles per hour for that electrical communication within the neuron. The important thing to remember about the action potential is that it is only triggered when the excitatory or go signals exceed the inhibitory signals. This is called the threshold. Increasing the stimulation above the threshold will not increase the neural impulse's intensity. In other words, whether or not a neuron fires is an all or nothing response. At rest, the inside of the cell is negative 70 microvolts. With inputs to dendrites, those receptor sites at the end of the cell body, the inside of the cell body becomes more positive. If the resting potential rises above the threshold, then action potential starts to travel down the cell body. So this figure shows the resting axon being approached by an action potential. So neuron to neuron. How do neurons communicate between each other? The axons branch out and end near dendrites of neighboring cells. The axon terminals are tips of the axon branches, and a gap separates the axon terminal from dendrites. This concludes the first part of the neuron presentation.